we know according to the nature of their chemical neurotransmitter. A little bit louder, please. That's for all the people. Because look, I will raise the same questions that you examine. Always the same. That's very important for you, I think, for your training, to listen to these questions and to listen to their answers. Okay, good. It's a little bit louder for everybody, but not for me. I know it's sort of.
Energetic neurons, energetic, paralytic, energetic neurons.
Good.
And there are some several of
Good. Good. Okay. I have another question. What is the difference between peptidagic neurons and monoaminergic neurons in terms of the synthesis of their specific neurotransmitters chemicals, monoamines and peptides? What is the principal difference? This is a repetition before you exam, right? Not very successful. Yeah, please.
I'm sure that I quite understand the question. Do you mean where they are produced?
Yeah, where they are produced, yes.
Well,
In at least in what compartment of the neurons? What kind of compartments of the neurons do we know?
Dendrites,
Dendrites.
cell body, and axons.
Absolutely.
And while peptidagic neurons are produced mainly in cell of the bodies.
In cell bodies. And also in dendrites.
In dendrites, yes. However, there are not that much fraction of them. But
And the monoamines.
monoaminergic neurons, they produce enzymes in any parts of the neuron.
Yeah. And therefore, monoamines themselves are produced all over the whole neuron, yes?
Yes.
In any compartment of the cell body. Yeah, that's a principal difference. Good. Now let's continue to. Yes. Yeah.
You mentioned during the previous lecture that peptidagic neurons are also synthesized in the axon, but only a small fraction. So it almost doesn't count, or
The small action.
a small fraction.
A small fraction. Peptidagic neurons. No. Peptidagic. There are much more. They are much more numerous than monoaminergic neurons. Well, so.
No. I mean that a small fraction of peptidagic neurons are also synthesized in the axon. No. No.
Look. According to basic data, theoretically, really, you are quite right. A very small number of peptides can be synthesized in reactions, but these are really traces of peptides. And I'm not sure at all that these peptides have a really functional significance. So it has been shown, but not from a physiological point of view. This synthesis is not important. Okay. Well, and the last question is, what is the construction? What are the compartments of the synapse? And what does it mean, synapse? What is its functional role? Yeah, please. Ladies first. Well, so what are the compartments of synapse? And what is the functional significance? And how does it function? Okay, good. Yeah, please.
So the compartments are presynaptic cell, synaptic cloud, and postsynaptic cell neuron.
Yeah, okay, good.
You can functions like the cell goes like to come in the cell between them and to create connection between the cells in order to have for learning informations.
Good. And what is the function in the molecular way? How it does function? If somebody knows? You know everything. you could replace me. I think.
Well, of course, but as far as I remember, the enzymes are produced inside a cell. Then, by some process that involves how they are contain vesicles, and then are moved to the synaptic clot. And after that, these enzymes are connected to the receptors on the postsynaptic neuron and also on the neuron that is themselves. And then these enzymes are connected to the receptors. They enhance some processes in the postsynaptic cell.
Good, okay. And now we will show this function. Well, so first, this slide, it's summarized. Well, so we will see the function of the synapse. Huh? Well, so the information is coming to the presynaptic terminal in terms of nerve impulse. Okay? Then there are vesicles which accumulate neurotransmitter molecules. Okay? And these vesicles discharge their content neurotransmitter molecules from the cells. While what kind of mechanism? How is it called? Exocytosis. Okay. Well, so chemical molecules are released from presynaptic terminal. Then they reach receptors on the surface of the postsynaptic membrane of the target neuron. And then what happens then? Well, there is a dissociation of chemical molecules and elimination from the intercellular place by specific enzymes. How do we call enzymes which eliminate peptides? Nobody knows. Peptidases. There is a lot of peptidases. And how do we call enzymes which eliminate monoamines? Monoamine oxidases. A and B. Okay, good. Well, so neuron in the human brain consists of up to 100 billion neurons. And there are each neuron receives up to 30,000 synapses. Well, so that is a very complicated regulation and exchange of information. You can imagine that each neuron receives an information from up to 30,000 other neurons. Good. And it integrates finally. Well, so, and there is an expression of 2,000 of signals, chemical signals, per second in the brain. Well, so there are many years ago, I mentioned already, that even in the 60s, there were great scientists which considered that we could reproduce the functioning of the brain, or in terms of our terminology today, this is artificial intelligence, okay, to reproduce. Well, so, but at that time, we knew only one chemical neurotransmitter, that was acetylcholine. Well, and it was possible at that time to do it, theoretically. But actually, after discovery of hundreds of thousands of chemical signals, it was evident that it is impossible to reproduce the functioning of the brain. And therefore, these great people, like academician Gilfan, they canceled to do it anymore. Okay? Well, and the brain consists of a lot of thousands or millions of networks which are involved at different levels of the brain in the regulation of different functions, in cognitive functions, in motor function, and in many other functions, in the regulation of peripheral organs like cardiovascular system, endocrine system, etc., etc. And the neurons communicate in between by synapses due to chemical signals. Okay? And this is very important because the information it is transferred from one neuron to another within this network. But I mentioned already, probably I mentioned, that these chemicals, they cannot provide their action in the periphery. Their action is limited within the brain. Why? Most of them, I mean peptides and monoamines, they are released in the intercellular cleft, but they cannot reach blood vessels. I mean, they cannot be released into the blood vessels and circulate. Why?
Maybe it's about blood-brain barrier.
Absolutely,
That's-
absolutely. Because in the brain there is a blood-brain barrier consisting of endothelial, continuous endothelial cells, without fenestra, without the windows, which are permeable for substances. Also, in the periphery, there are so-called fenestrated blood vessels with the windows, small windows, which are highly permeable for different substances. Well, so there is continuous endothelial cells. Moreover, in the endothelial cells in the periphery, where there is no blood-brain barrier, there is a lot of small vesicles which transfer substances from one side of endothelial cells to another side. And in the brain, there are only a few vesicles of this kind. And finally, this blood barrier is composed also of a shift of terminal processes of glial cells. Well, so they occupied all the surface, the whole surface of the blood. Therefore, there is a blood-brain barrier which is not permeable to all substances which are not highly dissolved in lipids, because all the membranes which should be permeable, they consist of lipids. And lipids are dissolved, highly dissolved in lipids. But that is not the case for proteins. Proteins and monoamines, they cannot be dissolved in the lipids. And therefore, for peptides and for monoamines and for most amino acids, the blood-brain barrier is not permanent. Well, good. Now let's come to the final question of this lecture. What is the role of the brain in the integration of the whole organism? Well, I like very much Dali, Salvatore Dali. Do you know his picture? No, probably some of you have been visited his museum in Barcelona. Yes, you have visited it. Well, so I like very much this painter because he was a really philosophical one. And therefore, I always, when I give a lecture in integrative physiology, I show this picture, which is called the Galatea of the spheres. Do you know what does it mean, this picture? No? Well, see, he loved for the whole life his wife, and that was not the case for his wife, on the contrary. But, he married, she was originally Gala. Her name was Gala. And he was originally from Kazan. And he made a picture. This is the picture of the pretty farm, which is composed from the elementary particles. Well, so the idea was how the riddle of that, how is created and maintained the internal harmony of his wife, and why it is broken. Well, so that is actually, this is the main problem of the integrative physiology. And what does it mean it is broken? When this integrative mechanism are broken, people, usually that is only attention or in the diseases. Huh? Well, so these people, they become very, very poor and they lose this integrated, because they lose this integrated mechanism. But in general, what are the integrated mechanisms? Or what are the mechanisms for the control by the brain of all the brain functions and all the peripheral functions? And there are two systems. Well, so in other words, brain is a conductor of neural and neuroendocrine regulation. Regulation, in other words, regulation of the brain itself and of the whole organism, including cardiovascular system, endocrine system, pulmonary system, etc., etc. Well, so first the information, and there are two major functions of any organism, of any body. Do you remember what are these crucial functions?
Sensory.
Which these functions, they give a guarantee of the survival of the organism. What are two functions?
Adaptation.
Sorry?
Adaptation.
Adaptation, exactly. Adaptation to the changes of the internal and external environment. Yes. Well, so, or in other words, this function is called a keeping or maintaining homeostasis. Okay. Well, and this is the main function. And another one is reproduction. If there is no reproduction, there are no new generations. Yes. Well, so there are two major functions, and these two functions, they're under the control by the brain. And first, brain receives the information from sensory system about the environment and about the external environment and the internal medium. What does it mean, internal medium? The composition of the blood, because blood is an indicator, the composition of the blood. This is an indicator of the function of the functioning of all the peripheral organs. Okay. Well, so then brain, it takes a decision, it gives an analysis of this information, and it takes a decision. What shall we do? Huh? And it passes his decision to internal organs, and it gives the brain, it gives an instruction for execution. What should be done to improve the situation, to improve homeostasis, etc., etc. And there are two principal systems, which are complementary. The first one, this is a nervous system. And the nervous system composes of nerves, of axons within the nerves, which innervate directly the internal organs, and they also integrate the functional activity of the brain. And the information is distributed here as nerve impulses. Okay? Well, so what is regulation? What is the characteristic of this regulation? It is fast, and it is short term. Well, so this action is the duration of this action is only during the association of chemicals with receptors. After dissociation of chemicals and receptors, there is no anymore the action of the brain on the peripheral organs. Okay? Well, so the regulation, fast and short term. And there is also tonic regulation for the whole time at a very small level, but this is a tonic. It is provided by so-called neuroendocrine system. A neuroendocrine system, this is a combination of the brain and endocrine system. And there is a hypothalamus. I will tell you in more detail next lecture probably, that in this region of the brain there is so-called hypothalamus. And the hypothalamus, it provides a lot of mostly neuropeptides and dopamine, which play a role of so-called neurohormonal factors, not as neurotransmitters, but neurohormonal factors. They are released into the local circulation, blood circulation, which connect, which is between the hypothalamus in this region and the anterior lobe of the hypophysis. And the anterior lobe of the, and in the anterior lobe of the hypophysis, there is a secretion of all the hormones which control the functional activity of endocrine glands. Well, so at this level of the hypothalamus hypophyseal system, there is an integration of the brain and the peripheral endocrine organs. Well, so and this regulation, I repeat once again, is slow and tonic. So this is a sort of a background of regulation. Good. And now let's go to the next lecture, the next topic. Let's say because this is what is the methodology and advanced techniques for brain research. Yeah, it is very important. Well, so I told you already that over the recent 30 years, there was a development of new paradigm, principally new paradigm for the study of the body, which is called translational medicine. Could you remind me, what does it mean translational? What is translated? From what you where? I explain you.
Science results.
Sir, yeah, please tell it for everyone, not for me. I know it exactly.
Yeah, translational medicine is about translation of science results to application in medicine.
Repeat once again.
Translation
Translation from what?
from science to application in medicine.
Not really. It's in a very general way of this one. Yes, you are right, but not. But you didn't say about the specificity of translational medicine.
Maybe translation is about translation of experience of curing one patient to general.
Well, look, I will explain you again. Do you remember that, I don't know, during the previous lecture or even earlier, I told you that there are several paradigms for the development of biology and medicine. There is preventive medicine, there is translational medicine, and there is, and the third one? Personalized medicine. Okay? Well, so preventive medicine is very old. It was developed since Hippocrates or even earlier. Well, but translational medicine, it has been developed over recent 30 years. What does it mean? That over recent 30 years, we have developed a lot of techniques, a lot of facilities to study all the events in the body at the molecular level, at the cellular and molecular level, not only at the level of the whole body and at the level of individual organs, but at the level of cells, of individual cells, and at the level of the molecules. And therefore we exactly know, or we can study, what kind of molecules are responsible for any physiological process, and what kind of molecules are destroyed or are modified, providing or provoking the pathological processes, the diseases. Okay? And these data on the molecular mechanism are translated into the development of new diagnostic and treatment technologies. Okay? Well, so if we know these individual molecules, we can use them as targets for the development of innovative techniques for the diagnosis and for the treatment. This is the principle of translational medicine. Well, so I return once again to this topic. Well, so I will explain you what is the methodology for the study of the cellular and molecular mechanism of the functioning of the brain, of the brain, and subsequently for the development of new technologies. Well. And there are at least three approaches to study the functioning of the brain. First, and we have used for that invasive and non-invasive techniques. Is it clear what does it mean, invasive and non-invasive? Okay, good. And there are several stages, several steps in the study of the brain. First, it is very interesting to study the functional activity of individual neurons. And now we have all the opportunity, all the facilities to isolate individual neurons from the brain and to put them and to keep them in so-called cell culture. Okay? Well, so we can study what are the syntheses here, what is the functional activity, what is the electrical activity, etc. So we can study individual neurons in cell culture, in dissociated. This is a dissociated cell culture. Okay? But certainly we know that all the neurons interact with many other neurons. Do you remember I mentioned already that individual neurons receive information at least for maximum 30,000 other neurons, up to 30,000. Well, and therefore, certainly we should go to the next step of the study, the interrelations between neurons in the networks, mostly within the synapses. And therefore, this we can do in so-called slices or in fixed sections. We can make with special equipment, we can make the sections, usually they're 300 micrometers thick, okay? And we can keep them in a special method. When we could change different chemicals, we could modify the functional activity of these neurons, and we could register what is the consequence, what is the functional consequence for the interaction between two neurons. And we can study exactly what happens within the synapses. Okay? Well, so first we'll make the slices like this, thick sections. Well, and finally, we can study everything at the level of the whole brain, in vivo, in animals. Also, we can make surgical procedures, we could make pharmacological procedures, and many others. But
Sorry. Question.
Yeah, please.
When we is it the problem that we slice the brain just for example in this way, but neurons can be interconnected in another way? Not
Yeah,
just
yeah.
the problem of course.
Yeah, sure. That you know that I told you that there are different steps for the study of the whole brain from individual neurons, but certainly they function in the networks, within the ensembles. Then we can study interactions between two or several neurons within the brain. But finally, we should study the brain functioning as a whole, with all the networks, with all the neuronal ensembles. And therefore, we can do it in animals and in human beings with special techniques, experimental techniques, and non-invasive techniques in people. Okay. Well, and let's see. And there are several techniques to study the functioning of individual neurons. Because all the mechanism of the functional activity of the brain in terms of neurotransmission, it started from the expression of genes which are responsible for the synthesis of specific enzymes, for monoamine synthesis, or for the specific neuropeptides as neurotransmitters. Well, so, and the expression of genes is represented by messenger RNA. Well, so messenger RNA, it is synthesized on the template of DNA. Well, so DNA, why, this is a double strand molecule, and it is dissociated. And then one of the strands is used as a template for the synthesis of messenger RNA. Okay? It happens, this procedure is realized within the nucleus of the neuron. And then messenger RNA with the registered information about the composition of the protein, it is released, it is delivered from the nucleus to the cytoplasm outside the nucleus, to the cell bodies or to the dendrites. Okay? And then this messenger RNA is used as a template for the synthesis of neuropeptides and of enzymes, for instance, of monoamine synthesis from so-called amino acids. Well, so messenger RNA and DNA is synthesized from nucleotides, and peptides are synthesized from amino acids. Okay. Well, so there are two techniques which give us an information about gene expression. One technique, this is a biochemical one, and we can study gene expression or synthesis of messenger RNA in the individual regions of the brain. Well, so we dissect this region of the brain, we make a homogenate, and we study in the homogenate the content of messenger RNA. And this technique is called PCR real-time, polymerase chain reaction. Okay? Well, but I told you already that any brain regions, they consist not only from one population of neurons, but from many of them. And therefore, in the homogenate, we will collect the peptide-adject neurons, monoamine-adject neurons, and many others. It is very different to differentiate what is the polymerase, what is the messenger RNA for peptide synthesis or for monoamine enzyme synthesis. And in order to study the messenger RNA within individual cells, neurons, we can use at sections the technique which is called hybridization in situ. Well, so we make a section, histological section of the brain, and we identify the neurons which are particularly interested for us, and we started to use this technique to start to locate and to stain specific messenger RNA in these cells. Is it clear?
Well, not really. So I didn't get difference between this.
What is the difference between
Those two methods.
No, so look, if we're interested in the estimation, same of gene expression, we should study messenger RNA.
Yeah.
Messenger RNA. Well, and we can study this by using two kind of different techniques.
Mhm.
Biochemical, when we started that in the homogenate of a local region of the brain. We dissected by scissors or by blades, razor blades, these regions, and we studied as called messenger RNA. But we could differentiate them, messenger RNA for one peptide or for another. But still, this is in the homogenate, which contains a lot of different neurons. Okay? But if we're particularly interested in the study of specific individual neurons, we should use this morphology. This is biochemical technique, and this is morphological technique. Well, so we make a slice or a section of the brain, and in a section we could identify specific neurons which are particularly interesting for us. Okay, and in these neurons we can use this technique which is called hybridization in situ, and we could stain specific messenger RNA for different peptides. Okay, it's clear? Good, let's go. Well, so, and I told you that the initial procedure of the functioning of the neurons in terms of the production of specific neurotransmitters, this is gene expression, messenger RNA synthesis. What is that? And then there is a synthesis of the protein. Of the proteins, either of neuropeptides, which play a role of chemical neurotransmitters, or the enzymes for monoamine synthesis. But all of them, they are proteins. And there is again, well, and these immunological techniques, they were developed earlier than methods for the estimation of expression of genes. That they were developed in 70s and 80s. Okay, what is the idea? What does it mean, the protein? Protein, this is an antigen, and we could develop against these antigens specific antibodies. Okay, and these antibodies, you understand what I mean?
Yeah.
Antibodies. Well, so we inject to the animals of this antigen, and in the body of these animals, it is shown. It is shown there. There is a production of antibodies against this antigen. This is an adaptive reaction just to eliminate these antigens from the body, which may be toxic for the body. Understand this? Right? Okay. Then these antibodies, these antibodies, they can recognize directly specific individual protein molecules. Very selectively. Well, so these antibodies, they are labeled with different stains, with fluorescent stains or with any stain. And after that, there are two techniques to visualize these proteins in the brain. Like in like with messenger RNA, there are two approaches: biochemical, and we use again a homogenate of certain brain regions. And in the homogenate, we use this technique, Western blot, or Western blotting. These are antibodies which are labeled with different stains, and which recognize in the homogenate specific proteins. And we could calculate what is the amount of this protein in this local brain region. This is a biochemical approach. And there is also a morphological approach, which is called immunocytochemistry. Well, so again, you make a section, the histological section of the brain, and you detect with these antibodies stains with fluorescent, you detect the proteins which are interesting for you. It's either neurotransmitters or the enzymes of monoamine synthesis or any other enzyme. Okay? Well, so this is a principle. Well, so primary antibodies, which recognize the proteins on the section, and then there is another antibody which is labeled with fluorescence. All the primary antibodies, they can be labeled with fluorescence right away. Okay?
So differences on the scale of this method.
What?
The difference is scalar. I mean that the Western blot is for the whole tissue.
Yes. For homogeneity.
And molecular chemistry is for several neurons.
That's right.
The whole population, several of them.
Look, if you use biochemical approach,
Okay.
it is called immunoenzymatic assay or Western blot. So almost the same. It means that you descend the brain regions, which is very interesting for you for its function. For instance, you study the brain regulation of motor behavior. You should dissect substantia nigra, which contains dopaminergic neurons involved in the regulation of motor behavior. If you are interested in the study of cognitive functions, you should dissect hippocampus and cortex, which are responsible for the regulation of cognitive memory, learning, social adaptation, etc. Then, in the whole homogenate, using these two techniques, Western blot and the monoexamine, you could select what are the proteins are produced in this local region. Well, okay, but hippocampus, as any other system, they contain numerous different neurons according to their phenotype, according to specific synthesis. And if you would like to differentiate in this homogenate in different neurons what are exactly proteins are produced there, you make a section, you find the neuron which is interesting for you, and you use mono or multiple labeling of different proteins. And you could detect it within these cells of interest, this neuron of interest.
Okay.
And this is immunocytochemistry.
So this technique allows us to differentiate the different neuron phenotypes.
Yes, exactly. Exactly. Well, so, and at the beginning of the developing of this immunocytochemical technique, it has been developed at the end of 80s. We can use only monoimmunolabeling. So we could recognize only one protein. Nowadays, we can use multiple labeling, right? Double labeling, triple labeling, etc. And we can recognize within the same neuron several proteins which are particularly important for the functioning of the neurons. Good, let's go. Well, and now I will give you just an example from our study. I told you also already that there are, do you remember? I told you that there are major neurons are represented by peptidergic neurons, which synthesize peptides. The monoaminergic neurons, which synthesize monoamines by using of several enzymes, specific enzymes. And I mentioned already that there are non-typical neurons. What are these neurons? Do you remember? Yeah.
Just several neurons users again.
Yes, this is the first one. There are neurons which can produce several neurotransmitters, even of different chemical nature. And we will listen to the presentation today at our seminar on the paper which summarizes the data on this topic. And there is another one, just an opposite possibility. Do you remember?
There is only one.
Monoenzymatic neurons. Okay, good. Monoenzymatic neurons, and we made really important contribution to this, to the study of these neurons. Well, look please. And this is double immunoleveling, immunocytic. This is the picture, huh? Immunocytochemistry. Well, so we labeled neurons, we stained neurons with different composition of enzymes for monoamine synthesis, huh? in the hypothalamic region. Well, look please. This yellow, this not sorry, yes, yellow. These are neurons which contain both enzymes for dopamine synthesis, huh? Tyrosine hydroxylase and aromatic L-amino acid decarboxylase. It means that these neurons, they can synthesize dopamine from A-tyrosine, from the amino acid precursor. Well, so they do not need any aid from other cells, okay? But at the same time, there are neurons, red neurons, which contain only the second enzyme of dopamine synthesis, aromatic L-amino acid decarboxylase. And only there are green neurons. And the green neurons, we can see here only axons of these neurons, huh? Green, which contain only the first enzyme of dopamine synthesis. And we have demonstrated, yes, and these cells, which contain only tyrosine hydroxylase and only decarboxylase, I will show you a little bit later, and I showed it previously, huh? They can synthesize dopamine in calculation. Well, so A-dopa, the precursor of dopamine, which is synthesized from L-tyrosine in these neurons, is transferred, is delivered to these neurons for dopamine synthesis. Yeah.
If I understood you correctly, those that respond with tyrosine, they cannot synthesize dopamine, so they transfer tyrosine to another neurons to produce dopamine.
No, so A-tyrosine, this is an amino acid precursor for any neuron, for monoenzymatic and for enzymatic, yeah? A-tyrosine. But A-tyrosine is synthesized in these neurons which contain only tyrosine hydroxylase. It is produced from L-tyrosine, L-dopa. L-dopa, this is an immediate precursor of dopamine. But there is no another enzyme, decarboxylase, to convert L-dopa to dopamine. And therefore, L-dopa is transferred, is delivered from this neuron to the red one. And in these red neurons, L-dopa is converted to dopamine. Understand? Well, so we have demonstrated at the morphological level, first in confocal microscope, that there are very close, very intimate relations between both cells. This is an indirect evidence of this transfer, and these intimate relations serve to facilitate the transfer of L-dopa from one cell to another. Well, so you can see this is confocal microscopy. This is the same red neuron, and this is the same action, huh? But in the volume. This is the three-dimensional demonstration. But still, at the level of the light microscopy, fluorescent microscopy, the resolution power is not enough to say that there is really direct contact between one neuron and another, because the resolution power is too low. Huh? And therefore, we have used electron microscopy. And with a much higher resolution power. Look, this is the axon which contains aromatic L-amino acid decarboxylase. It is dense. Huh? It is gray or even dark, okay? And this is axon which is in close relations. Look, there is no anything between these two axons. And this axon is labeled with silver grains. Huh? And this is a stain of tyrosine hydroxylase. Well, so we have demonstrated not only at the light microscopic level, but also at the electron microscopic level, that there are direct contacts between both mono and zymatic neurons. Clear? Let's go. Well, and I will show you how it looks like, synapse at the electron microscopic level. This is presynaptic axonal terminal, which contains dense core vesicles at the electron microscopic level. Dense core vesicles, and this dense core is represented by neuropeptides or monoamines. Okay, and then small so-called synaptic vesicles. Synaptic vesicles, they also contain neuropeptides or monoamine, and this is presynaptic terminal. And the accumulation of small synaptic vesicles at the terminal, in the local region of the synapses, at the membrane, is called active zone, active synaptic zone. Because the transferring of chemical signal from presynaptic terminal to the synaptic one is realized within this active zone. And you can see that there is a high density of presynaptic membrane and postsynaptic membrane within the synapse. Well, so this is a synapse, in fact, between an axon and the dendrite. Okay? Yeah.
But the vesicles are smaller,
Small vesicles, yeah, synaptic vesicles.
actually, so why that the size reduces when the vesicles closer to the synaptic?
Well, yes, you are right. Well, so look, it is believed that there are two pools for storing neuropeptides and monoamines. The stable pool, and these are dense core vesicles, no? And the labile pool, mobile pool. It means that when individual, for instance, nerve impulse is coming here, that at the first stage, at the first step, the neurotransmitters which are contained in these small vesicles are released. Okay? And after all, the neurotransmitters which are contained in dense core vesicles are released. But it is also believed that these small vesicles are generated from dense core vesicles.
So the big ones are like a warehouse.
Yes, exactly. Well, so okay, I will show you. Well, so this is a dense core vesicle, huh? And this is a small vesicle. Small vesicle. Well, so here and here, there is neurotransmitter inside, okay? But when there is an activation of the neurons, first neurotransmitters are released from these synaptic vesicles by exocytosis.
The big one.
Yes, but if there is a long-term stimulation, well, so these dense core vesicles, they begin to give rise to small vesicles, like this. You understand?
So they don't collapse, it just releases.
Yeah, yes, yes.
Wow.
Well, so the small vesicles are separated from dense core vesicles having inside neurotransmitters. And then neurotransmitters are released via exocytosis of these small vesicles.
Like the buds of the gist cells. Like gist, when they generate new cells, they also the buds. It's like similar.
Yes, very similar. You know, I already told you that the nature, the logic of the nature and of our mind is quite different. And usually the nature, it uses very simple mechanisms which are reproduced not only for the nervous system, but for any other cells and any other system. I mentioned to you that in the last year there was a huge congress on physiology in St. Petersburg, Russian. Unfortunately, only Russian because of the political limitations, etc. But, I gave a plenary lecture, and I told people that there are no neurotransmission, there are no immunotransmission within the immune system. There is no endocrine transmission in the endocrine system. Yeah? This is the schemes which are reproduced in our mind. But the nature, it reproduces the only one mechanism. The same chemical signals in their nature, these are peptides, monoamines, amino acids, which provide their action on the same receptors. The same chemicals and the same receptors are contained all over the whole organism. In the brain, in endocrine glands, in the immune system, etc. For instance, I will give you an example that we can see that there is a neuropeptide, gonadotropin releasing hormone, which consists of 10 amino acids, but it doesn't matter. Well, so this neuropeptide is synthesized in the brain. In the brain, and it controls the sexual behavior, for instance, not only, but the sexual behavior as a neurotransmitter. Then the same neuropeptide is released from other neurons in the hypothalamic region into the general circulation, and it controls the secretion of endocrine glands. Okay? The same peptide is synthesized in thymus, and it controls the functional activity of immune system, immunocompetent organs. Well, so, but in our mind, well, so in this Congress, there were different sections, different symposiums. Neurotransmission with chemical signals, chemical transmission in immune system, chemical transmission in endocrine system, etc. And they stated that there are no individual systems for intercellular communication. The nature it produces only one, using the same chemicals and using the same receptors. But the difference is that the functional effect, final functional effect of these cells is different. So in immune system, this peptide, for instance, it controls the secretion of antibodies. In the brain, they control the functional activity of the target neurons. In endocrine glands, they control the secretion of hormones by other endocrine glands, etc. But chemical signals and receptors are the same all over the whole body. Yeah, please.
So, is that correct? I understand this the same idea, for example, with serotonin expression, because it's a immune form, because it produces in the brain and in the gastric system.
Absolutely. Well, so serotonin, for instance. Moreover, in our one of the laboratories in our institution was created by a really outstanding physiologist. His name was Kashayans. Well, so at that time, serotonin, it is considered as a chemical, as a brain neurotransmitter, and that's all. But Kashayans, he expressed an idea that the same serotonin we could find at the very low level of the development of our organism, in the individual, in ontogenesis and in phylogenesis. And in fact, serotonin is produced at the very low, at the level of multicellular organism, or even at the individual, in the individual cells. And also at the very beginning of the development of fetuses, okay, of embryos. But serotonin is the same as in the brain in adulthood, in adult mammals, and receptors for its action are the same, but the functional action is quite different. In the brain, it plays a role of neurotransmitters. At this very low level of the development, it plays a role of of the chemical which integrate the functional activity of neighboring cells.
Mm.
Well,
Is it correct to call it neurohormone? Because I sometimes hear such terminology.
let's look. We say always hormone if it is released and it is distributed for a long distance. Why so-called body fluids? Well, so but at the level of two cells organism, multiple, there is no body fluids. Well, so this blood circulation or cerebral ventricles, they are developed at the most advanced stage of the phylogenetic development. Well, so we can say that this is a chemical signal, intercellular chemical signal, and that's all. Well, so I repeat once again that the nature is much more intelligent than we are, and it finds a very simple mechanism for the functioning of the whole body, because there is a maximum guarantee of the surviving of this organism, and therefore the signalization is the same all over the body. And only in our mind we separated it for neural regulation, for endocrine regulation, for immune regulation. In fact, it's the same. Well, so my colleagues, which developed this idea of, for instance, of immune regulation or endocrine regulation, they were very, very angry with me when they told that. Well, okay. Yeah.
Could you please clarify once again about the small vesicles and their spores? Because I don't understand, there are two types of vesicles. One is smaller and then spore is larger. And small vesicles are released first.
I will explain again, once again. Look, there are two pools. People believe, there are no two really absolute evidence, but people believe that within the presynaptic terminals, there are two pools for storage of chemical signals. There are dense core vesicles with the dense core, and there are small clear vesicles, synaptic vesicles. Well, so it is believed that dense core vesicles, this is a stable pool, while the synaptic vesicles, this is the level pool, which could erect very rapidly. But these two structures, they are interconnected because synaptic vesicles are raised from dense core vesicles.
Raised?
Yes,
I mean,
yes, look, like this. I wrote this. This is dense core vesicles, but synaptic vesicles can be separated, can be originated from the dense core vesicles. And some chemical molecules, chemical signals, they are captured by these synaptic vesicles. And after that, synaptic vesicles are separated, okay? And this is a level. Just at the beginning of the stimulation, of the neural stimulation, this level pool is released by synaptic vesicles, okay? Yeah, please.
But the hormones, you said that usually the hormone we need the regulator molecule that is transferred through the blood fluids.
Body fluids.
As I remember, this transport that there is a humoral regulator, so it's transferred through the blood, right?
But look, there is a discrepancy in terminology
Yeah.
in Russia and in Western countries.
That's
Because in Russia, we call humoral medium, humorality here, humoral medium, the blood circulation, cerebrospinal fluid, etc. We call in Russian humoral medium. But in Western countries, they call the same as body fluids.
okay, so this is a sign.
This is the same body fluids. Well, so body fluids is also the general circular blood circulation, These are cerebrospinal fluid, these are lympha, etc. etc. Well, also that's the same, but the terminology is a little bit different in Russia. And okay, good. Okay, let's go on. Good. Well, and now, if we return to the techniques for detection of monoamines and metabolites and monoaminergic neurons, we should say that there is a lot of, several at least, techniques to detect the functional molecules of these neurons. And there are several functional molecules. You remember that this is vesicular transporter, vesicular membrane transporter type 2. Dopamine and monoamines are synthesized all through the neuron. They are released by exocytosis. They provide the action on receptors, on autoreceptors, and they capture back by membrane transport. Well, sure, we can study first. There are different complementary techniques to study these neurons. In my laboratory, we have all of them. First, this is high-performance liquid chromatography. This is a biochemical technique for studying the content and concentration, let's say, of monoamines themselves or their metabolites in tissue homogeneity. Again, for instance, you are interested in dopaminergic neurons, nitrostriatal dopaminergic neurons. You dissect your substantia nigra or striatum, and in this region of the high concentration of dopaminergic neurons, you estimate the content of monoamines and metabolites. Okay. Then immunoenzymatic assay, you can make the same, but with another technique, with immunoenzymatic assay by using antibodies. This is immunological technique. But these are the same. The result is the same. And histofluorescent technique. The histofluorescent technique, because if you, with monoamines, after their fixation with formaldehyde, they become fluorescent. And this is histology. This 1 and 2, these are biochemical techniques, and these are morphological techniques. First, this is histofluorescent technique. This was the first technique that was developed for the detection of catecholaminergic neurons. It was developed in Karolinska Institute in Sweden. But now it is not used anymore because it's not very sensitive and not very specific. Now we use immunocytochemistry on sections of individual neurons of enzymes for monoamine synthesis and also of monoamines themselves. Double immunostaining, we could study not only the distribution and location of enzymes, but also the monoamines themselves in the same cell, in the same neurons. Okay? And we can use also isotopic technique to study, to detect monoaminergic neurons. Do you have an idea how we can use isotopic technique? What does it mean? So that, yeah, please.
The researchers put an isotope and using something like positron emission tomography, they detect the distribution of the isotope in tissue.
Yes, okay, but look, in local brain regions, there are monoaminergic, for instance, dopaminergic neurons, there are peptidinergic neurons, there are any other, and we should detect only dopaminergic neurons. You say that we could inject isotope, and but it was distributed all over the brain, all over the
We can
tissue.
connect this isotope to this dopamine. for
Yeah,
example.
and
And it will be used only where the dopamine.
why? That's right, but why?
Because it's used and consumed by the neurons that produce from this processor the dopamine.
No, it's not the case. What is the idea? Why? Let's show it quite correct. If you dissect slice, tissue section or cells, peptidagic, dopaminergic, etc., and you would incubate these cells with the tritiate dopamine, it will be captured specifically to dopaminergic neurons, not to peptidagic neurons, not to serotoninergic neurons. Why?
You are sensitive to.
Sorry?
They are sensitive to that particular molecule.
Yeah, I don't know exactly. Well, a little bit louder for everyone, please.
Because so, for instance,
Well, so look,
when
there is dopaminergic neuron, and there is peptidagic neurons. Well, and this tertiated dopamine, it will be captured only by dopaminergic neuron. Why?
I was thinking probably the family neurons, they have a special sensitivity.
Yeah.
Yes.
All right. But what is the mechanism of this procedure?
No.
You don't know. I repeated it several times. What is the specificity of dopaminergic neurons? It is drawn here.
Maybe for the for this you have antibody and
No, this mechanism is drawn at this picture.
Messenger.
Please find what is the mechanism. It is there. nothing difficult.
Okay, the question is why this dopamine is consumed by dopaminergic.
Exactly. Well, so there are two neurons.
Yeah.
Dopaminergic and peptidagic. Yeah.
Yes.
You incubate with tritiated dopamine, and only dopaminergic neurons will be captured by exactly by dopaminergic neurons, but not peptidagic neurons.
The receptors for specific.
What is the mechanism? Exactly. There is a specific uptake of dopamine. It is drawn here. This is the dopamine membrane transporter, which is responsible for capturing of dopamine, tritiated or labeled or non-labeled, etc.
So it's like vacuum cleaner for dopamine.
Sorry?
It's like vacuum cleaner.
Absolutely. So everything is drawn. I told you about that several times.
So just the reuptake.
Yeah, look, we incubate, well, so in peptidagic neurons, there are no transporter like dopamine membrane transporter, okay? Well, so we inject in this medium, we inject tertiated dopamine. It can be taken inside the neuron only by dopamine transporter. There is no another way to enter the neuron, okay? Good, understand.
And for do they use a dopa or dopamine?
Dopamine.
Dopamine.
Because this is for L-dopa, there is a membrane transporter but in any cell, and it is not specific. This transporter is valuable not only for L-dopa, but also for a lot of large amino acids.
So dopa can be consumed by other neurons and dopamine only.
Yeah, L-dopa can be taken up by almost any neuron. Well, so if we inject L-dopa to the medium with dopaminergic neurons, serotoninergic neurons, peptidagic neurons, etc., it will be included in all the neurons. Because all the neurons, like the majority of cells, of any cell, they have a transporter for amino acids, for large neutral amino acids, and this is a dozen or 20 amino acids, and L-dopa. And this is non-specific transporter. And here we have a absolutely specific transporter first, and the same transporter, specific transporter, we have for serotoninergic neurons and for the third one, dopaminergic, serotoninergic, and the third one,
Noradrenergic.
noradrenergic, or norepinephrine.
Because of them are.
Yeah, all of them, they have specific neurotransporters. Well, for their own neurotransmitter, okay? Well, good. Good. Well, so and now I okay, 1 minute. I just have time to tell you. But then we will next lecture we will start with evaluating with studying the non-invasive method for brain research. And there are two principal major methods, approaches, magnetic MRI and PET scan, positron emission tomography. And that is the development of this technique that was extremely important because now we have an opportunity and facilities to study the brain of humans. Well, so that is without destroying the brain, without injecting something, electrodes or some materials, etc. This was a revolutionary in the neurology and in psychiatry. Good. Now you will have an interval for 20 minutes, yes? And after that, we will have a seminar.
Those blocks are running forward.
Sorry?
Those blocks are running forward with the brain.
Okay. So please stay. Let's continue. I'm sorry. It was not intention. Look, please. Now, let's see. I raised your question previously. There is MRI, and most of you, or some of you at least, you use this MRI in your studies, or in the studies of your supervisors. But MRI consists of three different modifications: magnetic resonance, classical background, functional magnetic resonance, and magnetic resonance spectroscopy. Do you know probably what is the difference between two approaches? What kind of results we can obtain with different? Yeah, please, you're welcome.
We use magnetic resonance if we want to get structure of the brain. And
Exactly. Morphology,
Yeah, and function.
with a very good resolution, and there is a 3D dimension, there is a scanning in 3D dimension.
And functional magnetic resonance or MRI we use when we want to get some, say, sequence of MRI scans and see not on structure but on some functional changes in the blood signal.
It's not very clear what you said. Look, what does it mean function in memory? If someone is studied the function of memory, the technical assistant he says, please think about your wife, and he recognizes what is the region of the brain is actuated, or thinks about your very hard, very difficult professor like me. So there is another brain region which will be labeled. Okay. Well, so my question, and this is functional MRI. What is the background? What is the mechanism of the demonstration of this, or the demonstration of the labeling of these active zones? Yeah, please.
Well, as far as I understand, we try to detect the change in the concentrations associated in these regions. And the difference that we obtain is the difference in magnetic features of the associated proteins.
Exactly. Well, so in fact, we do not detect the functioning of the brain itself. We detect only the blood supply of these regions. And we believe that if the brain region is activated functionally, there would be an increased blood supply of this region. Okay? Well, so this is a detection of the blood supply and of the hemodynamic effect. Okay? Good. Well, what does it mean, magnetic resonance spectroscopy? Yeah.
Maybe it's about studying some chemical changes in the brain.
Right, right. And more concretely, could you give any example or idea of
Of some hormones, maybe.
look, the principle, you're right. Huh? Fundamentally, you're right. What does it mean? That with MRI, we could calculate the concentration of specific materials, chemicals, within the brain, in the local brain regions. For instance, in Parkinson's disease, there is an accumulation of iron, ions, within the substantia nigra. Ferro, yeah? You understand? Within the ferro, iron. But in the substantia nigra. If there is, and therefore this test can be used for the diagnosis of Parkinson's disease. If you see an accumulation of iron in the brain, in the substantia nigra, you can tell, even if there are no motor dysfunction here, you can say that there is a development of Parkinson's disease. Well, so this is one of the examples. I say once again that with this spectroscopy you can measure local concentration of different substances in different brain regions. Sometimes it is very important, like in Parkinson's disease, for the diagnosis.
Not the.
So
Further.
I didn't get the exact mechanism, so they know from, I don't know, post-mortem or something like that, the thickness, for example, of certain tissue, and you just apply some, I don't know, some special parameters for the MRI scanner, or how exactly is it different from magnetic resonance?
Yes. Yeah. What is an advantage of the use of MRI when comparing with pathologic materials? Yes, right? That's your question.
No. My question is the exact mechanism that differentiates this magnetic resonance spectroscopy, because I didn't get the exact mechanism for how do we I don't know, get that in a certain region we have something different with this tissue. There's
With MRI, yes, with MRI. Listen, I think that in spectroscopy, this is the same principle. There is either dissociation of a special light, or I think spectroscopy. This is the same for any time. With spectroscopy, you can use you can study the spectrum of different substances, their compositions, etc.
Okay.
Well, okay. Other questions? My dear friends, may I have a rest? It's you. Welcome.